Okay, good morning. Yesterday we were talking about weed characteristics. So we want to move on to other issues. Okay, let me admit somebody. Right. Just a recap on what we were doing yesterday. Yesterday we focused on weed characteristics. And we referred to Becker, I think in 1975, who listed about 12 probably traits which describe weeds. Then we expanded on these traits, getting more examples about these weeds. So we should know the characteristics of weeds which separate them from other plants. This is what we covered yesterday. We also tried to define the weeds. Weeds. Let me admit someone. Okay, I'm trying to admit some people. Okay, we define the weeds as simply vegetation which we find in agricultural or let's say in cropping fields. So it's simply vegetation which we find in agricultural fields. There's also an evolutionary way to look at weeds or to define weeds. These could be other plants. Or these could be ruderals. Or competitive plants. Remember, there are two approaches. So today what we want to look at are three things. So today we want to focus at three areas. Number one, we want to look at weed seed dispersal. This is simply the movement of weed seeds. The other thing we want to look at is weed seed bank. Then we also want to look at weed seed dormancy. So let us start with this weed seed dispersal. Then probably around the 12 or yeah, we have finished. Yeah, morning. Alright. Ah, because I didn't want to read. Zvirikutoticha.
Alright. Iyi ndeyadr Lugari yakasiyana nechienzi yakuda handizotoendesa
Okay. Bhodhi racho riri kuitwa rini?
na10:30.
Ndiri
Alright.
kutozopedza masikati.
Masikati. Saka indokusira
munogona kundisira muningi
muenvelope
ndova ndichitakura.
muchiita alright.
Hello, sorry for disturbance. So I was saying we are going to move on to talk about the seed dispersal. So with the seed dispersal, what is it? This is the movement of weed seeds from the point of production to other areas. So this is basically the movement of weed seeds from the source of production. The source of production is actually the plants themselves, which we call weeds, which have gone into flowering, then they go into seed production. And you have a seed movement and dispersal mechanisms. Now, the causes of seed movement are basically three. We have got abiotic and biotic, then agronomic. Abiotic consists of the action of wind and animals. Sorry, wind and rainfall. This is the abiotic. The biotic involves the animals. So we will start with wind. The term which is actually used is anemochory, to refer to wind effects. Hydrochory, this is the movement of weed seeds due to rainfall. Biotic causes, which are classified as zoochory, this involves organisms which are capable of dispersing seeds by adhesion. Seeds can stick to the animal skin or even to human clothes. This is epizoochory, or it could be through ingestion, endozoochory. Now, dispersal carried out by ants is not very large. It's classified as myrmecochory. Then there is a self-dispersal where you have a splitting of the fruits of the plant, which is known as autochory or ballistic dispersal, which involves barochory and pseudo-serotin. These are just terms which are used. Then anthropochory, this involves humans which act as fundamental drivers of dispersal. We have a horizontal seed dispersal mechanisms. This is what we call anemochory, or just wind seed dispersal. This depends on wind speed. If you don't want to use these terms, just use the simpler term wind, or if you want, you can use anemochory. Most of the weed seeds in Zimbabwe are likely to be dispersed by this method, by wind, during the dry season. For example, like this current period, we could have wheat seeds which are being moved around by the wind from place to place. A result of wind-driven aerial movement can actually redistribute wheat seeds. You need special type of wheat seeds with certain features. These wheat seeds resemble parachutes. For example, Conaiza species. Some wheat seeds can have wings or plumes that aid flight. So what you are saying is that for the wheat seed to be carried by the wind, it should have certain features which allows it to float in the air, like a parachute, like a feature, or it could have some wing-like structures which can aid flight. This is Conaiza arbida with those fluffy seed heads. Actually, they have parachute-like structures. Agimon mexicana. Let me admit somebody. Sorry for that.
Doc, on this side, ndichiri paslide rakanzi biotic causes. I'm not seeing the weeds you are mentioning.
What about others?
Yeah, I can see Agimon mexicana.
Yeah, someone is seeing, someone is at the right slide. Is it not your system there?
Yeah, maybe my system or the network is poor.
What about others? What are you seeing? Hello?
I'm seeing Mexicana.
Okay, so I think it's your system.
Yeah, okay, let me try to use YouTube maybe.
Yeah, if you go to YouTube, I am live there.
Okay, Doc.
Yeah. Okay, so we're talking about features which can aid floating of these weed seeds in the air. Milenis repens is also seed heads which can easily be carried by the wind. Now we move on to the effects of water, hydrochory. Now water can actually transport weed seeds around. At the onset of rains, of course this may involve some erosion. So we have got seeds which are transported by water runoff or by floating along water drainage channels. It could be irrigation channels. This form of dispersal is likely to be common during the rainfall season in Zimbabwe, probably around, should we say, around November when we get the rains. So weed seeds capable of floating in water are transported to other areas. Or we can have a splash seed dispersal by raindrops. Let me check, there could be somebody again who wants to join. Right, raindrop action, but this is to a very small extent where you have the movement of the seeds by raindrop action. Epizoochory, this is very common where you have seed adhesion to animal fur. Seeds may have certain features which enable them to stick to the animal skin. So the animals which are involved, things like cattle, horses, donkeys, sheep and goats, probably cattle, donkeys and sheep, goats, which are commonly used in smallholder farming areas, which can roam about the fields as they graze. Like during this period, they also move around with seeds. To a certain extent, wild animals are also involved in this type of spread of weed seeds. So the seeds should have special characteristics which make them attached to the animal skin. Let me admit someone again. So I was talking about, seeds which can get attached to the animal skin as the animals move around. They spread these weed seeds. The examples of weed seeds which are adapted to this type of dispersal, we have Xanthium strumarium. Then we have got Bidens pilosa. We have got Tribulus terrestris. We have got Acanthospermum hispidum. Xanthium strumarium seed. This is what it looks like. It has got those hooks. And it can easily get attached to the animal skin. I'm sure it can cause, because it has some thorns, I think it causes some irritation. and the animal will try to remove it, and during the process the seed is dispersed. Tribulus terrestris seed. This is common in, I think, natural region 3. You can see those spines. It can pierce somebody or animals. And of course, the reaction is to remove it and throw it away. It becomes dispersal. Biden spirosa tsine, you know, it has got those hooks. And if you move in a field with this weed where infestation is high, you end up with a lot of seeds on your clothes. Same applies to the animals which move through such fields. Acanthospermum hispidum seeds, it also has those spines, and it can aid this weed to be dispersed by sticking either to animal skin or to clothes. Setaria verticillata seed head. Again, this one can be very difficult to remove when it sticks to your clothes or animal skin. It's a grass which is common in agricultural fields. Endozoochory, we are now talking about seeds which are eaten by animals. These could be birds which eat the seed. For example, Thomas in 1970, who was studying the biology of Rotbolia coccincainensis at the weed research team, he found that guinea fowl and cattle, livestock, were involved with the dispersal of Rotbolia coccincainensis. Because these birds, guinea fowls, could eat a lot of seed. Cattle also grazed this grass. Rupend also found that Leucine indica was associated with the manure application. So cattle could aid the dispersal of Leucine indica. This one is a minor dispersal, minor coccory or dispersal carried out by ants. The distances involved are probably 2 to 5 meters. And we have Datura stramonium seeds, which are known to be dispersed by ants, as reported by Ben Venuti in 2007. Now, these seeds have got a lysome. It's a chemical which can attract ants, and in the process, the ants end up distributing these seeds. Ballistic autochory is where we are having a fruit which explodes or which splits and it disperses seeds forcefully from the fruit. Maybe you know about this. You might have seen plants which can split, especially during this period. In fact, it's common with some trees with pods. They can actually crack and drop some seeds. This also same applies to some weed species. But short distances could be involved around the mother plant. Datura stramonium can also do this by splitting its fruit and broadcast the seeds around the plants. Cliomonophylla mujakari spindle pod. It has got elongated pods which when mature they can also split and scatter the seeds around the plants. What is known as barochory is not a true dispersal mechanism because of the absence of any device for moving the seed away from the mother plant. Seeds actually remain inside the plant. They are in an open calyx in the apical zone, or they are just inside the fruit. This may prevent seed predation. Examples are Chenopodium album or Amaranthus erythroflexus. This is Chenopodium album. Then another term known as pseudoserotin. Probably this may not apply to the actual weeds. The phenomenon of seed retention in the closed, usually woody fruits or cones. Seeds are protected from also predation. These are other plants, and probably they may not be applicable to weeds. Humans are involved with a lot of seed dispersal. This is known as anthropochory. Agronomic activities, working with implements. You have seed adhesion to the tires of operating machines. It could be clothes or even animal skin of draft power animals. Combine harvesters used on several farms could end up, dispersing weeds from farm to farm. Coming. Yeah. The lecture probably after 12. Sorry about the disturbance. So I was talking about combine harvesters which are used on several farms, which can end up dispersing weeds. We also have application. Let me check something. Right. Now, application of manure could spread the weeds, such as Elucine indica and Amaranthus hybridus. This was reported by Rupende in 1997. strike acid can contaminate crop seed, and if we sell that seed due to seed trade, we end up spreading these weed seeds. Actually, the presence of Chenopodium arubum, which we find today in Zimbabwe, came through wheat seeds from Europe. Now, seed movement within the soil is very limited. We can have a seed burial depth, which is linked to germination. It affects buried seed availability. That is mediated by phytochroma in photosensitive species. So we are talking of this vertical movement. So deeply buried seeds may actually be affected by lack of gases, a term which we call hypoxia, or inadequate gas exchange. This can actually inhibit germination. Soil tillage operations can also influence the distribution of weed seeds, but within the soil profile, this is very limited movement. Minimum tillage systems, we have the weed seeds which are concentrated near the soil surface. However, conventional tillage where we use the plows could result in uniform mixing of weed seeds in the soil profile. But on heavy soils which crack, the cracks could facilitate the movement of weed seeds down the soil profile. There are some weed seeds which can bury themselves in the soil. For example, wild oat, it has got a certain structure which, if it is wet, it can uncoil itself and drive the seed inside the soil. Probably you are aware of certain grasses which do the same thing. Burial also accounts for the weed seed longevity and dormancy, which we will talk about. We have talked about weed seed longevity. We also have the activity of earthworms, which can bring buried weed seeds to the soil surface, but these are very short distances which we are talking about, and which are happening within or in situ in the same field. A similar weed seed movement has been attributed to beetles, so insects can move around weed seeds within the soil profile. Ants also act as agents of weed seed vertical movement, but this is not movement to great distances. So the vertical movement is also it is also happening within weed seeds. If you want more information on weed seed movement and the dispersal strategies in the agricultural environment, you may have to read this reference, Weed Biology and Management. It is well written, you may understand the movement of wheat seeds. And with that, we come to the end of this wheat seed dispenser. I'll stop streaming. Yeah, do you have any questions on wheat seed dispenser? Actually, the reason why we have a lot of weed problems is that they can easily move from one place to the other. And there are a lot of agents which are involved within this movement. This is the reason why we always get weeds in agricultural fields. It's partly explained by weed seed movement. Right, we'll move on to weed seed banks. Remember, when weeds are formed, weed seeds are formed, they end up in the soil and they are stored there for a long time, and it may depend on the longevity of each weed species. Some may last for 4 years, which is a short span, but others can last for 20 years. So let us talk about weed seed banks. Coming. Hello.
Yes, someone was.
All right. Even 6 seconds. All right. Okay, we now want to talk about weed seed banks. Now, weed seed bank is actually the soil where most of the seeds will reside after shedding from the plants. From various studies, we now know that these weed seeds can remain for a long time. No wonder why we always get weed problems. There are some scientists or agriculturalists who wanted to take or who wanted to take this route of exhausting the weed seed bank so that we have a weed-free environment. This is quite a difficult task, and it may not be possible to create a weed-free environment which is permanent. So where are the weed seeds stored after formation and shedding? Most of the weed seeds are stored in the soil. No wonder why we have the term soil seed bank. Some could remain attached to the plant, and we can have aerial seed bank which is very small. So what is a seed bank? After weed seed shedding, seeds are deposited on the soil surface. This is where we find them. The weed seeds could be incorporated into the soil during tillage operations. Seeds on the soil surface and in the soil form what is known as the seed bank. Seeds may also remain attached on the plant. This is the aerial seed bank, but this is not much. So we can have a variety of weed seeds on the soil surface. When the conditions become favorable, the seeds will actually germinate. And give rise to seedlings. Now this is shamba grass. At the base you can see, probably you can see the seeds which are scattered after shedding. Now the weed seed banks are near the soil surface. If you dig the soil deep down, you may not find any weed seeds. So here I've got a schematic representation of what can happen to weed seeds. Let us start with the green plant that weed. If it flowers, it can produce seeds, and they are dropped to the ground as seed rain. Once they are in the soil, probably they will go into innate dormancy, induced dormancy, or enforced dormancy. They go in a state of dormancy and they are stored within the soil. Now, we also have weed seeds which are coming from elsewhere through dispersal. This is what we have been talking about. We spent a bit of time talking about the dispersal mechanisms which can bring in new stock of seeds into the fields coming from somewhere. Probably this looks like indirect investment. Then we have predation. Predation, you have got animals which can eat this seed. Some seeds may attempt to germinate, but they will fail. Some may be affected by pathogens. Some are at deep burial. And some are physically damaged. And some may actually become dead. That is the fate of these weed seeds. Probably this could be a better diagram where we focus on weed seed bank dynamics. Now, if you have got weed seeds which are in the soil, they are in two states. It's either they are non-dormant seed bank or a dormant seed bank. Non-dormant seed bank, you can easily withdraw if it was real money, in a bank, this is liquid money which you can easily withdraw. But some monies are fixed, dormant. You cannot just go there and withdraw them. So non-dormant seed bank can give rise to germination. After germination, some plants can actually die and not produce seed, but others which can produce seed through the seed rain or a dropping of the rain of the seeds, the seed can enter into the system. Or you can have a seed which is coming from far away via rain, wind, animals, machinery. Seed dispersal, which we have actually covered, can contribute to the seed banks. Then you have decay. These are withdrawals of seeds from the seed bank. Pathogens, fungi, predation, you have got the birds, mice, and the insects. Why we refer it to as a bank? This actually mimics the actual monetary banks which we have. That's why we are using this sort of a model so that we can understand what is happening to the weed seeds as if they are everyday monetary transactions. So what is a weed seed bank? We have got the reserves or viable weed seeds present on the surface and in the soil. They consist of new seeds shed by the plant or older seeds in the soil for several years. So the seed bank is an indication of the past and the present weed populations. There are enormous numbers of weed seeds in the soil. In fact, it is the soil where the seeds are stored. So some weed seeds may eventually die within a few years, but others may persist for a long time. This is weed seed longevity. Excuse me, let me check something. We will continue. Let me check a certain message. Come in. Yeah. Yeah, yes.
How are you today?
Yeah. The lecture is
What was the lecture?
right now. So I do suppose that my 12.
Okay.
Yeah.
That's fine. Thank you.
All right.
You got a lunch. What do you do for lunch?
Yeah, after 12, then that is.
Oh, so I'm not
Eh?
What will you be having for lunch? Are you having lunch? Is two different things.
Yeah, sorry about that disturbance. So let's move on to talk about the density of weed seeds, which are linked to the cropping history of the land. Now, weed seeds may actually reflect what has been going on in a cropping field. So, seed density is actually influenced by the past farming and may vary from field to field. Thompson and Grime in 1979 divided the seed bank into two groups. The first group, they referred to it as the transient seeds, where you have grasses which are mostly dominant in the weed seed bank, which do not persist for more than 1 year. The second group is what is typical of the cropping fields, the persistent seed bank, where you have species of a wide range of habitats. These are the weeds in agricultural fields. Now in grasslands, the majority of the seed bank is located within 2 centimeters of the soil surface, and nearly the entire seed population is located in the 15 centimeters of the soil. In conservation agriculture systems, the seed banks move closer to the soil surface. Seeds are in a better position to germinate and interfere with crop production. But under good weed management, the seed bank could be more easily reduced in conservation agriculture. Now let us look at the tillage effects on the weed seeds. Now tillage can affect the vertical distribution of seeds in the soil. In undisturbed conditions, the weed seeds remain near the soil surface. The example is the conservation agriculture or reduce the tillage. And the tillage with the moldboard plow buries the weed seeds deeper, probably to depths where they become dormant. And the reduced tillage can keep the weeds, the weed seeds close to the soil surface, like in conservation agriculture. This can reduce dormancy and encourage germination. And indeed, in some conservation agriculture systems, you find a lot of weed seed, a lot of weed infestation. The reason is that we have plenty of weed seeds near the soil surface, which can easily germinate. Here we have got examples of weed distribution in the soil profile under different tillage systems. Now, in moldboard plow, in conventional tillage, you get almost sometimes uniform distribution, or you get less weed seeds on the top, but you have plenty of them at the bottom. I am not going to talk about chisel plow, but what is common here is the moldboard plow and the reduced tillage, CA. In CA, it tends to be the opposite of conventional tillage, where you are going to have more weed seeds at the top and less as you go down. Now, there was a study which was done by Mavunganidze et al. In 2009. They found that the 0 to 10 centimeters depth had higher weed seed numbers than the 10 to 20 and the 20 to 30 soil depth levels. Probably it depends on the tillage which was being done in that field. It looked like reduced tillage. Now, fields which had been cultivated for a long time, more than 20 years, had larger seed banks than the recently opened fields. So the longer the field is under cropping, the more it is likely to have a lot of weed seeds in the soil compared to newly opened fields. So the soil acts as a medium of seed storage and growth for weedy plants. Now we want to look at the loss of seed from the soil. Now, seeds are eaten by rodents and insects. That's how we lose the seed. Or it could be through decay due to microbes. They may germinate and emerge. Certain experiments referred to as the Bureau experiments have indicated that there are some weed seeds which can last for 120 years and can still be viable, which are stored in the soil. This can indicate that weed problems are actually perpetual. It's something which we have to live with it. The Duvero experiments also indicated that some weed species could be viable up to 39 years. So seed longevity increased with all weed species, with all species, as the depth of burial is increased. So plowing action can bury weed seeds and they can be stored for a long time. And this seed longevity depends on the depth of seed burial, the amount and intensity of soil cultivation, crop management, and weed control programs can actually change or influence the weed seed bank size. Soil seed bank can be reduced by applying herbicides. Probably later on I will talk about how we can actually determine the weed seed bank size. Currently we have got 2 students who are trying to determine the weed seed bank sizes in agricultural systems or cropping systems in Murewa and Mutoko. Now, withdrawal of herbicides leads to increase of the seed bank to its original size within 1 year. So if we were using herbicides, we may manage to reduce the number of weed seeds in the soil. But the moment in which we remove the herbicides or stop using herbicides, the fields can revert back to their original status of high weed seed banks. How do the seeds enter into the soil seed bank? Or how are weed seeds deposited in the weed seed bank? The source of seeds, it could be weeds that are allowed to mature to produce seed. Weed seed production of standing weeds can contribute to the amount of weed seeds in the soil. Seeds can enter a field from outside sources, and the primary seed dispersal methods could be wind, water, animals, and humans, which we have already discussed. Surface irrigation water is also a potential to distribute weed seeds. Now, crop seeds which are contaminated by weed seeds, for example, wheat seed can be contaminated by Datura stramonium seeds in this country. Sorghum, pearl millet, and finger millet seeds are also contaminated with the weed seeds. And in fact, farmers do tend to share these crops. During the process, they end up, distributing weed seeds. Equipment movement can also distribute weed seeds. So many weed seeds can be eaten by animals and pass through the digestive tracts, but still remain viable. If we use manure, cattle manure, we end up distributing these seeds. Also, wildlife spread these seeds. So this is how they can come into the weed seed bank. Now, the germination and the emergence can be a form of weed seed withdrawal, because some weed seeds are actually used during the process, but it can also tend to be an investment. Should the plants become mature, they will produce a lot of seeds. So seeds can also remain dormant. We've got seed dormancy, and we've got 3 types of dormancy. That is innate dormancy, induced dormancy, and enforced dormancy. I mean, later on we'll talk about these terms. But I may mention now that innate dormancy is actually inherited. These are embryo status and the resistance of seed coverings to water and gas exchange. Induced dormancy is the germination does not occur even when the conditions are favorable. Enforced dormancy, these are environmental conditions, low temperature or lack of moisture, oxygen and light, which stops germination. Now let us look at the depth of barrier. And this can affect the germination and emergence. So the weed seed may remain stored in the soil. The ability of seeds to emerge from various depths depends on the soil and seed weight. Large seeds are able to emerge from greater depths, but we must also know that light penetration in the soil is affected by the silt, clay loam, and sandy loam soil, and maybe light can penetrate up to 22 millimeters. So for those seeds which require light, they may be affected by the soil types. And the depth of barrier can actually affect light penetration. It is actually light which can trigger germination or not. So let us look at the factors which can affect seed bank size. The total number of seeds within the soil seed bank is determined by a number of factors which can include previous history of the land, that is weed control, weed species composition, weed population, tillage, frequency, dormancy characteristics of weed seeds, and lifespan, longevity. Management of weed seed bank. How can we manage the seed bank? We can try to reduce the input of seeds into the seed bank via herbicide use, crop rotation, tillage, mulching, and also try to minimize weed seed dispersal. Probably this diagram may show all the factors which can affect the weed seed bank. So on our right, where we have this seed rain, we have got the seed bank, and we have got insects, fungi, and birds. We have also seed germination process. We have got dormancy, and we have got seed dispersal, bringing in the seeds. But on the other hand, we have got crop management. Things like mulch, fallow, intercropping, all of these human activities, which can actually influence this root, this blue root. We can actually reduce seed deposits into the seed bank by these human activities. We can also, actually we can increase seed deposits. Then we can also withdraw a lot of seeds because of these activities. But meanwhile, all of these processes are regulated by the agroecology, soil type, rainfall, and temperature. So these are the key drivers of weed density and diversity. Now I'm going to explain two methods of determining a soil seed bank. One method is popular, the other one is not. You will find out soon. The popular method of determining the weed seed bank is the weed seed germination method. What you do, you go into a field with an auger, which could be 5 centimeters in diameter, and take soil portions from 0 to 10, and put them in a bag. Take another 10 to 20, put in a separate bag. 20 to 30 in another separate bag. Now, after assembling the whole field, probably you need about 100 sampling points, which could be a lot of soil. However, increasing sampling will also increase precision, but there's a cost to it. How are you going to carry that soil? So the soil cores from the same depth are bulked together and passed through a sieve. The soils are then spread in asbestos trays. Then the trays are watered as necessary. What you simply do is to count weeds which are emerging, identify them, and record them. You pull them and remove them from the trays, and you have to wait until you get more weed seedling emergence. You can also disturb the soil after 1 month to encourage germination. You can do this for 3 months so that you have a reasonable number of the, you have reasonable numbers, or you can, you can do it for a long time in order to exhaust the weed seeds. This method was done by Mamunganidze in 2009, determining weed seed banks in Mzarabani area in Zimbabwe. There are 2 students who are also using this method currently, working in Murewa and Mutoko, trying to understand the weed seed banks in those areas. This one is the difficult method, the second one, because there's a choice. This one is called the direct seed extraction method. So what you do after sampling your soils, the weed seeds have to be separated from the soil using what is known as the flotation or sieving methods. This can be a messy process. Soil samples could be soaked in solution of sodium bicarbonate or sodium hexametaphosphate, sodium hexametaphosphate or sodium bicarbonate for a short time. You need a jet of water to remove clay and silt particles. Once seeds have been isolated, they can be identified. So you should be able to identify the weed seeds to their species level, which can also be difficult. Viability tests will also be done using the tetrazolium tests to determine whether the seeds are alive or dead. Probably you can have a look at this paper which determined the weed seed bank in a maize field. In maize fields. Anyway, these are the data from that paper. You can also use these references where these studies were actually done. Other papers as well on seed banks, and this is it on soil seed banks. Stop streaming. Hello?
Yes, hello.
Someone is raising. You want? Yeah. Ask me if you can. Is it Tendai? Hello, Tendai. Do you want to say something? Or your hand is, up accidentally.
Maybe it's accidentally because the microphone is off.
Hello, Doc. Sorry, my microphone now was off and I raised my hand by mistake. Sorry about that.
No, if you have got a hand up, I mean, it's still okay. I would think you wanted to ask me something, which is okay.
Hello, Doc. Sorry about that.
Yeah, no.
I raised my hand by mistake.
Okay. Zvakafana muri mukirasi kabura akasimudza ruoko unoti kuti yes. Yeah, so tapedza 2 areas. We've just done what? Seed banks. We did movement of wheat seeds. These are not they are not difficult sections. And last, we want to talk about. Do you want a break or you want me to continue?
Continue talk.
Because breaks are expensive if you are online, isn't it?
Yes.
Right. Let me continue. Probably we may finish early. Yeah, okay. We want to talk about weed seed dormancy. Now, weed seed dormancy is like a resting stage. Right. If we have crop seeds and put them into the soil after planting, we expect, if there's enough moisture, we expect the crop seed to germinate and emerge. But with the seed from the weeds, some will germinate and some may remain dormant. They may not germinate. Now this failure to germinate is what we simply call dormancy. The germination will happen later after a resting stage. So what is dormancy? It is a state in which viable seeds, it could be spores or buds, which fail to germinate under favorable conditions, such as moisture, temperature, and oxygen. Everything is there, but the process will not take place, the process of germination. So dormancy is actually the failure of viable seed to germinate under favorable conditions for growth of the seed. So here we've got seeds of Datura stramonia. Some may germinate, some may not. This is the germinating seed. When you see the radicle coming out of the seed and the plumule also coming out of the seed, then we have germination. These are the parts of the seed where we have the radicle which will actually protrude first. Then the plumule will also emerge. So what is the germination process? It is the resumption of embryo growth And if you look at these seeds, inside there's the embryo. So the embryo is the resumption of embryo growth and the emergence of the seedling from the seed. Process of initiating growth in seeds, resumption of growth of seed of any vegetative part. This can be regarded as germination. The process requires oxygen, temperature, right temperature, and sometimes light is required. In the case of photoblastic seeds, we can have live seeds in the soil, but they may not germinate, and we refer to these seeds as dormant. So, what are the types of dormancy? Actually, these were defined by Harper in 1975, who said that some seeds are born dormant, and that kind of dormancy is known as innate or primary dormancy. You have the seed which is already dormant, when it is produced, and that dormancy is innate or primary dormancy. But some seeds, when they are produced, they can germinate, but they will acquire that dormancy. That is what is known as induced dormancy. But some seeds may have that dormancy forced upon them. It's known as enforced dormancy. So there is innate dormancy, there is induced dormancy, the one which is acquired, the one which is forced or enforced. That one is known as that is an enforced dormancy, the one which is forced or thrust upon seeds. So induced and enforced dormancy constitute what is known as secondary dormancy. Here I've got a schematic representation of the three types of dormancy. So at the top we start with what is called seed dormancy, which we can divide into two: secondary dormancy and the primary dormancy. Primary dormancy is inborn or innate dormancy. The seeds, when they are born, they are already dormant. But with this secondary dormancy, we have got two types: induced dormancy and enforced dormancy. I think let us stop here so that we'll explain these three types of dormancy. So when we talk about wheat seeds, they can have three types of dormancy: inborn or innate, number one; number two, enforced dormancy; number three, induced dormancy. This is why the seeds can remain in the soil being viable for a long time. Now, let us focus on the types of dormancy. The seeds from a single wheat species may exhibit one or more types of dormancy, or all the three in succession over a period of time. This is for survival. We can use this example of a weed seed, which can alternate, types of domains. Let us start from seed dispersal. Now, from seed dispersal, you can have a plant with innate domains. It's not going to germinate because of inherited domains from the mother plant. Now, after seed shed, let's suppose we are in winter under cold conditions. The seeds will undergo stratification, let's say around June, July, somewhere thereabouts, when we have low temperatures. This stratification will change the ratio of inhibitors or hormones and cause the seed to break its domains. So we follow that seed. After the land has been disturbed, then it can actually germinate in summer. And more seeds are produced, and they continue to follow this cycle. But in some cases, the seed can go into induced domains, and it will fail to germinate. So it will cycle. There's no seed production here or germination because it's undergoing induced domains. But somehow it may end up in the outer circle and follow the normal process, or from here it may be temporarily go into enforced domains, which are caused by environmental conditions which may not be suitable. But when those conditions are removed, germination can occur and it joins the normal cycle. So to speak, what I am saying is that the weed seed can change its domain status. Either it follows the normal cycle, where it can break its domains, form germination process, produce new plants, or it can undergo enforced domains temporarily, then later join the main cycle and produce plants. Or it can circulate under induced domains without producing any plants, but somehow it may come out of it and follow the outer cycle and produce plants. So these changes ensure that the weed seeds don't just germinate, and it means survival for the weed seeds. I think that is the message which we want. Now we are going to talk about the innate domains. This is the domains when the seed is produced by the mother plant, which is the wheat. And we are going to spend more time on the innate domains, because that is the domains which is prevalent within the wheat. Now, primary or innate domains is the domains which is inherited from the parents. It is genetically controlled, and the seeds will not germinate if exposed to the environment which is conducive to germination. So innate dormancy or primary dormancy, it's inherited from the parents and it's genetically controlled. And because it depends on the genes. Even if the conditions are favorable, there's no germination at all. So what does it involve? It could be through morphological or physiological factors which cause this type of dormancy. So we are going to look at the morphological characteristics of seeds as well as the physiological characteristics of seeds, which are genetically controlled. There are some seeds which, when they are shed, they possess immature embryos inside the seeds. And for those seeds to germinate, the immature embryos have got to go, they have to grow and become mature so that the seed can germinate. We call this period an after ripening period for the embryo to mature. The examples of such weeds could be Xanthium strumarium, which can produce seeds which are just dormant. It can also be Acanthospermum hispidum, which can also produce seeds which are not mature. They will become mature after seed shed. Now on Xanthium strumarium, you can have two types of seeds. We call it polymorphism, where they produce morphologically and physiologically distinct set of seeds which have different after ripening periods. It's in Xanthium strumarium or cucurbillae. Yes, there is this after ripening period, but it varies among seeds, and we call it polymorphism in the Xanthium strumarium. But in the Acanthospermum hispidum, there's no variation at all. So this is the Xanthium strumarium, which can produce different types of seeds. In fact, if you crack that shell, you may have two seeds with different germination characteristics. This is for the survival of this weed. Now, after talking of after ripening period, there's also what is known as embryo dormancy. This is the failure of the mature embryo. This is actually a mature embryo which is supposed to germinate, but it will fail to germinate. Now, embryo dormancy is controlled by cotyledons, and if you want germination to take place, you may have to cut off the cotyledons. Then the germination process can occur. This is common in this plant known as Fraxinus species. But I don't have a good example for local weeds. Probably we may have to look for it. Now, embryo domains may also be controlled by a number of substances or hormones. Ethylene can stimulate the germination of several weed seeds. In Avena fatua, ethylene levels increase just before germination. So, ethylene levels within seeds can regulate germination in Avena fatua. We talked about stratification or low temperatures or chilling. This chilling process can increase a hormone known as gibberellic acid. Once that hormone is increased, it can actually affect germination. Gibberellic acid stimulates degradation of food reserves in the endosperm or cotyledons, which is an important process in germination. So you want the high levels of gibberellic acid or gibberellins to cause the germination process. Abscisic acid is also important. Abscisic acid deficiency is associated with the absence of primary domains. So if we have a lot of it in the seed, it can cause domains. So ABA is synthesized in the embryo and endosperm. It is then the balance between gibberellic acid and the abscisic acid which can determine domains in weed seeds. If you want a lot of detail about seed dormancy, you can read this journal. So dormancy or germination process can be a lot of processes as indicated by that diagram. And there's no need to remember this, but probably to read that paper and understand how these things are linked. Here we are looking at embryo dormancy, which is non-hormonal. Other non-hormonal compounds like phenolics, unsaturated lactones, short-chain fatty acids, and coumarin. These are other substances which can inhibit the germination within certain species. So if you want germination to take place, you have to remove these substances through leaching and oxidative destruction of these chemicals. If it is oxidative destruction, it could be a natural process within the seed. Then it can cause a breaking of the domains. Now the outside coat of the seed can also impose domains. we call it the tester. The seed dominance in many species is imposed by structures which surround the seed. This could be the tester, pericarp, bloom, palea, and lemma in cereals. For example, in Avena fatua, the palea or lemma, it is responsible for coat-imposed dormancy. This is the coat. So this can prevent entry of gases or water. But if you can pierce the coat, you can induce germination. So this is the seed of Avena fatua. Avena fatua seed. We are not really interested in the parts or naming of this, but here we just want to show that it is the outer part which is very strong,